Hello, and a very warm welcome to all of you listening in.
Hello.
Whether you are a seasoned postdoc currently pipetting away in a tissue culture hood, or, just someone insanely curious about the absolute mechanics of how life begins, we are really glad to have you with us.
It is great to be here. We have a particularly heavy-hitting topic on the table today.
We really do.
It's one of those subjects that really forces you to rethink what what you thought you knew about biology.
Absolutely. Today we are going to discuss the absolute ground zero of development. We're stripping everything back to discuss the very origin of life and really the scientific quest to capture the most powerful cells in existence.
And we're not just talking about stem cells in the way you might usually hear about them.
No, not at all. Not regenerating a knee or fixing a heart valve. We are going deeper. We are diving into the elusive search for totipotent stem cells.
This is not an exaggeration. This is the holy grail for developmental biologists.
Right.
When we talk about totipotency, we're talking about cells capable of creating an entire organism completely from scratch.
The whole thing.
The whole thing. Not just the body, but the entire life support system required to bring that body into existence. It's like the biological equivalent of the Big Bang.
That's a great way to put it. And to guide us through this, we are conducting a journal club on a fascinating major review article titled, let me get it right, Exploring the Origin of the Development Totipotent Stem Cells.
It's an incredibly thorough piece of work. Really comprehensive.
It is. This research comes from the lab of Dr. Peng Du at the School of Life Sciences, Peking University. It was published very recently in Current Opinion in Genetics and Development, specifically the 2025 issue, and we have to give credit where it's due. The lead authors on this review are Xi Li, Hui Shen, and Bing Peng, alongside, of course, Dr. Du.
Yeah, and before we really get into the weeds, it's so important to contextualize why we're even looking at this paper right now.
Okay.
This isn't just a summary of what's happened in the past. It synthesizes a massive shift in stem cell biology. For the last, 40 years, the field has been completely dominated by pluripotency.
Right. Pluripotency. We've heard that word for decades.
We've gotten very good at it.
Mm-hmm.
But this paper highlights the shift toward totipotency, a state that has been historically almost impossible to stabilize in a dish.
Okay, let's unpack that immediately, because I think for a lot of people, myself included sometimes, stem cell just means magic cell that could become anything.
Right, the catch-all term.
But you're saying there's a strict hierarchy here, a ladder of potential that we need to respect.
There is a very strict hierarchy. Biology is rigorous about this. You can't just skip steps or lump them all together.
So before we get into the results of the paper, which are, honestly, they're mind-blowing, let's lay the groundwork.
Let's do it.
For decades, as you said, the gold standard has been embryonic stem cells, or ESCs. So I guess the intuitive question is, why isn't an embryonic stem cell enough? If an ESC can become heart muscle, brain tissue, and skin, what are we actually missing? Why do we need to go back even further in time?
It's a great question, and it really cuts to the core of mammalian development. Think about a fertilized egg. That is the ultimate start point.
Right, the zygote.
Right.
The single cell that starts it all.
Correct. Now, that single cell, it has to do two completely distinct but equally important things.
Right.
First, obviously, it has to create the embryo, the fetus, the baby. That's the part we all focus on.
Sure.
But secondly, and this is just as vital, it has to create all the extra embryonic tissues.
Extra embryonic, so stuff outside the embryo.
Exactly. It has to build the placenta and the yolk sac.
The life support system.
The life support system, exactly. I mean, you have to remember, without the placenta, there is no pregnancy. There's no nutrient transfer, no waste removal. There's simply no baby.
It's the engine and the fuel tank. The embryo is just the passenger.
That's a good way to think about it. Now, here's the catch. As that fertilized egg starts to divide, it goes from 1 cell to 2 to 4. It hits a decision point very, very early on.
A fork in the road.
A fork in the road. It separates itself into 2 distinct lineages. One group of cells becomes what we call the inner cell mass. That's what makes the fetus.
The passenger.
The passenger, right. The other group becomes the trophectoderm, and that's what makes the placenta.
The engine.
The engine.
Yeah.
And where do you think embryonic stem cells come from?
They must come from that inner cell mass, the part that makes the body.
Precisely. So by its very definition, an embryonic stem cell is pluripotent. Pluri, meaning many. It can make every part of the body, but it has lost the ability to make the placenta.
So it's already past that fork in the road. It has already made a choice.
It's made a commitment. It has specialized. even if just a tiny bit.
I see. So if we use like a construction analogy,
Mm.
an ESC is like a general contractor who can build the entire house, the walls, the plumbing, the electrical, all of it.
Yeah.
But they can't build the foundation or the power grid connecting that house to the city. That work has already been subcontracted out.
That is a very, very fair analogy. And totipotency is the state before that fork in the road.
The master planner.
The master planner. A totipotent cell, like the zygote or the cells in the very first few divisions, which we call blastomeres, retains the potential to generate the whole new organism.
Both the baby and the life support system.
Both. It hasn't chosen a path yet. It can still become anything. It holds all the cards.
And that is what this paper from Peking University is exploring, how to capture that specific, all-powerful state in the lab.
Yes. And the stakes here are so high. Because without capturing totipotency, we can't truly model the very first days of life. We can't fully understand how a pregnancy establishes itself or, why so many early pregnancies fail, often before the mother even knows she's pregnant.
Right, that's a huge area of clinical need.
A huge black box. So the goal of the research reviewed in this paper is to, as you put it, freeze that fleeting moment in the lab so we can actually study it.
Freeze that fleeting moment is a pretty poetic way to put it. But, I mean, biologically, that is just incredibly hard, right?
It's been almost impossible.
I mean, have you ever just stopped to wonder how a fertilized egg actually knows it needs to start doing all this? It's just a ball of chemicals,
Yeah.
and then suddenly, boom, it's an organism. How?
Well, that boom has a name. It's called zygotic genome activation, or ZGA.
ZGA. That sounds like a launch code for a rocket.
It practically is. Think about the moment fertilization happens. When a sperm enters an egg, the resulting cell, the zygote, is initially running on autopilot.
Autopilot.
Yeah, it's using maternal factors, RNAs, and proteins that were preloaded and stored in the egg by the mother. The new genome, this unique combination of mom and dad, is silent.
Wait, it's sleeping. So the cell is dividing, but the new DNA instructions aren't actually being read yet.
Exactly. It's running on the mother's cache, if you want a computer analogy. It's coasting. ZGA is the alarm clock. It's the moment the new embryo's own genome wakes up and starts transcribing its own DNA for the first time. This is an absolutely critical milestone. If ZGA doesn't happen correctly, development just stops, dead in its tracks.
In this paper, it discusses how these totipotent cells are defined by markers related to that specific awakening event.
Yes, ZGA is the molecular signature of totipotency. It's the key that starts the engine.
Okay, so we have the definitions down. Pluripotent is the body only. Totipotent is the whole package body plus placenta.
Right.
And ZGA is the engine starting up. Now, let's get into the deep dive of this review, because the methods scientists are using to actually get these cells,
Yeah.
they're really clever.
They're innovative, to say the very least. It's some seriously elegant molecular biology.
The review starts by giving us a bit of a history lesson. It mentions the discovery of two cell-like cells, or 2CLCs, and this was back in 2012. This seems to be where the field really started getting some hope.
Right. This was the first hint that we could even access this state in a dish. Researchers found that in a normal culture of embryonic stem cells, and remember, those are the pluripotent ones.
The ones that have already chosen a path.
Right. In a dish full of those, a tiny fraction, like less than 1%, would spontaneously revert. They'd go backwards.
To a state that looked like a two-cell embryo.
Exactly. A two-cell embryo is very, very early. That's definitely totipotent territory.
And they had markers for this. They could see it happening.
They could. These cells suddenly started expressing specific markers like ZSCAN4 and a transcription factor called DUX. And ZSCAN4 and DUX are classic signs of that ZGA event we just talked about. They are the signature of the genome waking up.
So it was like a few cells in the dish were having their own personal ZGA moment.
That's a perfect way to describe it. They were rebooting themselves to an earlier state.
But there was a problem with these two CLCs, wasn't there? The paper describes them as metastable. That doesn't sound ideal for a lab experiment.
No, it is not. It's actually the opposite of ideal. And this brings us to a crucial concept in stem cell biology. Metastable basically means they are transient, they are unstable, they flicker in and out of existence.
Okay, let's try to visualize this. We use the bridge method here, because metastable can sound a bit abstract for our listeners.
Good idea.
So imagine you are hiking, and you're trying to balance a heavy ball on the very, very sharp peak of a mountain.
Okay, I'm with you. A ball at the absolute summit.
You can probably get it to stay there for maybe a second.
Mm-hmm.
That's the metastable state. It's high energy, it's totipotent, it's at the top of the developmental potential landscape.
It has the potential to roll anywhere.
Exactly. But gravity, or in this case, biological programming, is constantly trying to pull it down. It inevitably and very quickly rolls down one side of the mountain back toward the valley of pluripotency.
That is an excellent visualization. The two CLCs are that ball on the peak. They appear, but they do not want to stay there. They inherently want to differentiate or revert.
So you can't build a stable cell line out of them.
You can't. They keep rolling down the hill.
And for research, that's just a nightmare. You can't run experiments on a cell type that disappears every time you blink.
No, you need a stable population to do sequencing, to do drug screens, to do anything useful at all.
Exactly. You need a population that stays put. And that is where the big breakthrough described in this Peking University paper comes in. They describe a new type of cell, TBLCs.
Totipotent Blastomere-Like Cells.
And these are different from those unstable 2CLCs.
Fundamentally different in their stability. And the way the Du Lab and others achieved this it's just fascinating. They used a technique called spliceosomal repression.
Okay, spliceosomal repression. That sounds intensely technical. Let's break that down.
It is, but the logic behind it is beautiful and almost counterintuitive. They used a chemical inhibitor called pladinylate B, or PLAB for short.
Plague.
And this chemical, PLAB, directly interferes with the spliceosome.
Okay, let's bridge this for everyone. The spliceosome, that's the cellular machinery that it cuts and pastes RNA, right?
That's right.
It takes the raw instructions coming from the DNA and edits them into something usable.
Correct. DNA gets transcribed into pre-messenger RNA, which is full of coding parts, exons, and non-coding parts, introns. The spliceosome's job is to cut out the introns and stitch the exons together to make the final messenger RNA that goes on to make a protein.
So it's the editor of the cell's instruction manual.
It's the master editor, yeah.
So let's use another analogy. If the cell is a computer running a very complex operating system, let's call it pluripotency OS, the spliceosome is like the processor that's constantly handling all the active, specialized tasks.
Go on. I like this.
If you inhibit the spliceosome with this PLAD chemical, it's like hitting system restore or force quitting all the complex programs. You are jamming the very machinery that allows the cell to run its specialized software.
That is a very, very apt analogy. By dampening the spliceosome's activity, you're not telling the cell what to do, you're just stopping it from doing what it's currently doing.
You're breaking its routine.
And in that confusion, the researchers found, it essentially forces the cell to reboot. And when it rebooted, it didn't just go back to the complex pluripotent state, it defaulted back to an earlier, simpler state.
The totipotent.
The totipotent state, the factory setting.
That is incredible. So that by breaking the machine just a little bit, you reveal its original programming.
In a sense, yes. The paper notes that this inhibition reprograms the cells. And here's the key difference. Unlike the two CLCs, which are just, visiting that totipotent state for a moment.
The ball on the mountaintop.
Right. These new cells, the TBLCs, they stay there. They are stable. They can self-renew. You can grow dish after dish after dish of them.
They've built a plateau at the top of the mountain.
That's it. They've stabilized that high-energy state.
And the paper mentions they look different molecularly, too. It's not just about stability.
They do. This is really important. Remember we talked about ZGA, the genome waking up?
Yeah, the launch code.
The unstable 2CLCs express ZGA genes like ZSCAN4, but these new, stable TBLCs, they express pre-ZGA genes.
Pre-ZGA. So they're even younger in developmental time. They're from before the alarm clock even goes off.
In some ways, yes. They express markers like PID1 and CTE. These are genes that are active even before the embryo's own genome fully takes control.
So it's an even more fundamental reset.
It seems to be. And importantly, they have silenced the pluripotency genes. In the 2CLCs, the pluripotent software is still kind of running in the background, which is why they're unstable.
They're conflicted.
Exactly. In TBLCs, that software has been quit completely. They're a much purer form of totipotency.
This is a massive, massive leap for mouse research. But the whole point of this really is to get to humans. The paper also discusses the Holy Grail, doing this in human cells.
And that is where things get Even more complex, but also so much more exciting. The review categorizes the human counterparts into a bit of an alphabet soup.
Okay.
We have HCLCs, ZLCs, and HTBLCs.
Let's break those down because this is the absolute cutting edge, and I don't want anyone to get lost in the acronyms. First up, 8CLCs. That stands for 8-cell-like cells.
Right. These were identified by other groups. They appear spontaneously in cultures of human stem cells, just like the mouse 2CLCs.
And they express markers you'd find in an actual 8-cell human embryo.
Exactly. Markers like ZSCAN4 and DUX4. Note the capitalization, that's the convention for human genes.
So these are basically the human equivalent of those unstable mouse cells we talked about first, the ball on the mountain.
Roughly, yes. They are a transient population enriched with those CGA markers. But then
comes the spliceosome inhibitor.
Then comes the spliceosome inhibitor. The Door Laboratory, the authors of this review, identified a different population using that same chemical, PLAB. They call them ZLCs.
Zygotic-like cells.
Zygotic-like cells. That implies a single-cell state. That is very aggressive terminology.
Or at least very close to it. The paper notes that ZLCs are induced by that system restore method we discussed. And crucially, just like the mouse TBLCs, these human ZLCs silence the pluripotent genes completely.
They shut down the body-brumming program entirely.
A clean break. And the third type, the HTBLCs.
Human Totipotent Blastomere-Like Cells. These are also generated using splicing repression, but they seem to be the stable long-term version that can be passaged, meaning grown and multiplied in the lab over 20 times.
And that is the key, isn't it? Being able to actually grow them, that's what makes them useful.
It is the gold standard for utility in a lab. If you can't grow them, you can't really study them effectively. The HTBLCs also have reduced levels of a specific epigenetic marker called H3K27E3.
H3K27E3. Gesundheit.
It's a repressive mark on the DNA. Think of it like a molecular padlock on a book.
Okay.
Low levels of this padlock are a signature of the very early embryo. It means the DNA is open for business, it's ready to be programmed, the library is open, and all the books are accessible.
So we have these cells, we have the TBLCs in mice and humans. We've established they're stable, they have the right molecular markers. The big question is, what can they actually do? The paper mentions bidirectional developmental potential.
And this brings us right back to our fork in the road. Remember, pluripotent cells can only go down one path, the embryo path. Totipotent cells should be able to go down both the embryo path and the extraembryonic path, the placenta.
And these TBLCs, they can do this.
The data indicates yes. The review highlights that functionally, when you coax them to differentiate, HTBLCs can differentiate into embryonic lineages, making the building blocks of the fetus.
Right.
And they can differentiate into extraembryonic lineages, making the building blocks of the placenta. This is the functional proof. This is what shows they are, at least by this broad definition, truly totipotent.
They can build a house and the foundation.
The whole construction project.
And this capability leads to one of the coolest applications mentioned in the paper, blastoids.
Yes, blastoids, the synthetic embryo models.
It sounds like something out of a sci-fi movie, but it's actually a synthetic embryo, right?
Yeah.
Model of a blastocyst.
Essentially, yes. A blastocyst is the ball of about 100 cells that implants into the uterus around day 5 or 6 of pregnancy. A blastoid is a model of that structure created entirely in the lab from stem cells without using a fertilized egg.
And why are these new TBLCs so much better for making them?
Well, previously, scientists tried to make blastoids using other types of stem cells like extended potential stem cells or EPSCs, but the success rate was really low. The structures were messy.
A bit disorganized, like trying to build a really complex Lego set, but you've lost half the instructions.
That's a great way to put it. The different cell types didn't sort themselves out properly, but the paper notes that when you start with TBLCs, the blastoids they form have around an 80% success rate.
80%. That is a huge, huge number compared to what we used to see.
It is a complete game changer. And it's not just the number. They have better structural fidelity. The inner cell mass-like part and the trophectoderm-like part are in the right places. They look more like the real thing. This means we can generate thousands of these synthetic embryos to study early development, screen drugs for toxicity, or understand genetic defects.
All without needing to source actual human embryos, which is ethically fraught and logistically very, very difficult.
Exactly. It's the system restore paying off. You reset the computer to its factory setting, and now it reboots perfectly into the operating system you want every single time.
It gives us a standardized, scalable model.
Precisely.
Okay, so this all sounds like a victory lap for the field. We found the cells, we stabilized them with this clever chemical trick, and we can build better model embryos. But this is a scientific review. It's never that simple.
Never. Science is always messy. There's always a but.
And there's a whole section in this paper dedicated to the criteria for evaluating totipotency. And reading it, it feels like there's a bit of a debate simmering under the surface here.
Oh, there's definitely a debate. A big one. And it centers on the very definition of the word totipotent. It seems simple, but biologically proving it is an absolute nightmare.
The paper distinguishes between a strict definition and a broad definition. Let's talk about that.
Yes, the strict definition is the ultimate test. It's the goldest of gold standards. Can a single cell, all on its own, generate a whole new functional living organism?
So like if I put one of these mouse TBLCs into a surrogate mouse mother, is a healthy, viable mouse pup born at the end of it?
Exactly. And in mice, this has been proven for true totipotent cells like the zygote and early blastomeres. But for these cultured cells, the bar is incredibly high and the efficiency is still very low.
And for humans?
Well, for humans, it's impossible to test.
It's impossible because of ethics.
Right. We cannot, and we absolutely should not, be attempting to clone humans or generate pregnancies from lab-grown cells. The international guidelines are crystal clear on this.
So the strict definition is ethically off the table for any human research. We can't run the ultimate test. It's a non-starter.
So the field has to rely on the broad definition.
Yes, and the broad definition is what we've been discussing. It asks, can this cell differentiate into both lineages in a dish? The embryonic and the extra-embryonic.
Right. Or can it contribute to both lineages in something called a chimerism assay?
A chimerism assay.
Mm-hmm.
That's where you mix the cells into an existing embryo and see where they go, right?
Correct. You take your human TBLCs, you label them so they glow green or something, and you inject them into a mouse embryo. Then you let it develop for a little while and see if the green cells end up in the fetus and the placenta.
But the paper mentions a big problem with that, too.
Yeah.
It calls it interspecies chimerism.
It's the square peg in a round hole problem.
Okay.
Human cells do not like growing inside a mouse embryo. They grow at different speeds, they have different signaling requirements, different adhesion molecules. The whole environment is foreign.
So even if the human cell is truly totipotent, it might fail the test just because it's in the wrong species. It gets outcompeted or rejected.
Exactly. It could be a false negative. The cell is perfectly capable, but the environment is hostile.
So it's incredibly difficult to prove true human totipotency. We're relying on molecular markers and in vitro differentiation, they're good proxies, but they're not the strict proof.
And we are inferring potential rather than proving it through full live development.
And speaking of those molecular markers, there's another discrepancy the paper brings up that I found really interesting. It's this battle of the genes between the HCLCs and the ZLCs in humans.
Ah, yes. This is a critical point for the specialists listening in. We have these two candidates for the human totipotent cell.
The spontaneous HCLC and the induced ZLC.
Right. And they seem to run on completely different software.
Different gene networks.
Totally different. The 8CLCs, the ones found spontaneously, they rely on transcription factors like DUXA and LUTX.
Okay, DUXA and LUTX.
But the ZLCs, the ones made by the spliceosomal inhibition method from the D lab, they rely on a different set of factors, DDIT3 and DLX25.
That seems odd. I mean, if they are both supposed to be modeling the same stage of human development, shouldn't they use the same genes to do it?
You would absolutely think so. And this raises a really important question for the field. Which one is the real model?
Right.
Or are they just capturing slightly different millisecond snapshots of that ZGA process? Or is one of them an artifact of the culture system?
The review does suggest that the ZLCs might be more homogeneous.
Yeah.
Right? A cleaner population.
Yes. The 8CLCs are often described as dual-featured. They express the ZGA genes, but they also still express some pluripotency genes in the background.
They're confused. They haven't quite let go of their past life.
Exactly. They haven't made a clean break. The ZLCs, on the other hand, the ones made with plabs, seem to have made that break. They have fully silenced the pluripotency genes.
So the ZLCs might be a purer signal, even if they are artificially induced by that chemical shock to the spliceosome.
That is the argument. But it leads to a broader, more philosophical question that the paper touches on, which I think is fascinating. It's the artifact question.
Oh, I love this part. Are we creating a true copy, or are we creating a laboratory artifact?
It's a huge question. Think about the zygote in the body. Does it self-renew?
No, not at all. It divides once and becomes two cells. Then those two become four. The zygote state is gone in, what, a few hours?
Exactly. In vivo, in the body, totipotency is a transient bridge. It exists only to be crossed. It's a fleeting moment. But in the lab, these TBLCs self-renew
Right. They divide and make identical copies of themselves.
indefinitely.
So by their very definition, they are doing something that a natural embryo never, ever does.
So we have forced them to pause in a state that is meant to be temporary.
Correct. This is a fundamental biological difference, and it's caused by our culture system.
It's like pausing a movie. The characters on screen are frozen in midair, maybe in the middle of a jump. That state exists on the screen, but it's not natural for the character to just hover there for 3 weeks.
A perfect analogy. We have created a biological pause button. Now, is that pause cell identical to the one in the movie that's still playing? No, because the one in the body is dynamic. It's moving forward.
But is it a useful model? Absolutely. It might be the only way we can study that single frame of the movie in enough detail.
That is a really helpful way to frame it. It's not about achieving perfect replication of reality. It's about achieving accessibility. We are compromising reality just slightly to gain the ability to study it at all.
Precisely. And in science, that is so often a trade-off we have to make. A model is, by definition, not the real thing, but a good model is useful.
So we have covered the ground zero of development, this wild splicing hack that lets us pause time, and the very real debate over whether these cells are the real deal. Let's zoom out. What does this all mean for the future? Where does this take us?
Well, if you want to summarize the main takeaway from this whole body of work, it is that we have successfully moved the goalposts back. For 40 years, the starting line for stem cell biology was pluripotency.
Right.
Now, thanks to techniques like spliceosomal repression and the work of labs like Dr. Dew's, the starting line is totipotency.
We have rewound the tape all the way to the very, very beginning.
And this rewinding is what opens up what the paper calls the blastoid revolution.
The idea of using these cells as ideal seed cells.
Right. Think about it. If you want to study early pregnancy loss, which affects a huge number of couples, you can't easily study it in patients. It happens too early. And you can't use real human embryos for large-scale drug screening. It's just not feasible, ethical.
But if you can grow TBLCs in a vat,
You can generate thousands, even millions, of standardized, high-quality blastoids. You can screen drugs that might cause birth defects. You could screen drugs that might prevent miscarriage.
you could finally start to understand infertility mechanisms that happen before a woman even takes a pregnancy test.
Exactly. It allows us to open up the black box of early human development. For a long time, human development from day 0 to day 14 was a complete mystery. We just couldn't see it. Now we can build models of it on a massive scale.
The paper also mentions organoids. We usually think of organoids like mini brains or mini livers as coming from pluripotent cells. Would starting with totipotent cells be better?
The paper speculates that yes, it could be. Because totipotent cells have an even cleaner slate, they might generate organoids with less variability from batch to batch. They're the ultimate blank canvas.
It really is remarkable. We are essentially hacking the fundamental operating system of life.
We are, and we're doing it with this surprisingly crude tool. We're just jamming the processor, the spliceosome, just enough to make the whole system reboot. It yields a very sophisticated result from a relatively simple intervention.
I want to leave our listeners with a thought here, because we've talked about rewinding cells, we've talked about synthetic embryos, we've talked about capturing the moment of creation in a dish.
It definitely leads to a provocative question.
It does. If we can now stably culture cells that have the potential to become an entire organism, both the baby and the placenta, are we approaching a future where biology is no longer limited by the supply of eggs or embryos?
That is the ultimate implication, isn't it?
Are we moving toward a world where the only limit is our ability to biochemically rewind any adult cell? I mean, if you can take a skin cell, turn it into a pluripotent cell, and then use this method to turn it into a TBLC and then make a blastoid, the lines between body cell and embryo aren't just blurred anymore.
They're erased.
They're erased.
It suggests that embryo is not just a physical thing, but a software state, a set of instructions. And if we have the codes to rewrite that software, the possibilities and, the very heavy responsibilities that come with them are infinite.
A software state. That is something to mull over on your next commute.
Indeed, it changes how you look at every single cell in your body,
Thank you so much for breaking all this down with me today. It's heavy science, but it's truly defining the future.
My pleasure. It is a fascinating time to be in this field. There's never a dull moment.
And thanks to all of you for listening. This has been The Deep Dive. We'll see you next time.